thanks everybody for sticking around this long. Before I get started, I mean, most people in talks these days start out with a disclosure. And what that's all about is, well, who are you working for, and who's giving you money? And might that influence what you say in a public talk? And I want to actually state that I do have a disclosure to make, and it does influence what I do, and it does influence the way I think. And the groups that are involved in that actually are the National Institutes of Health. In other words, you, the taxpayers, who fund my work because you pay taxes, and the NIH is nice enough to give me some money in grants. Denny Sanford, who has provided funding for us to do independently what we want to do. And I think the most important thing is, of course, all of these funds that you see listed in front of you, like the Rocket Fund and Mason's Hope and the Veronica Marino Memorial Fund. These are individual families who have gone out of their way to give us money because they believe in the work and believe that we'll make some benefit. And that's what our obligation is to, because we don't work for anybody else, we work for the families. And as such, that's who ultimately I think we need to be in front of and talk about and talk to. And so I think the best way that we can get the best bang for the buck, if you will, is through collaborations. And that means collaborations that involve people all over the place, not just within our own institutions. And so that's why I've talked about this as the collaboration in glycosylation disorders here in La Jolla. And really, we've got two stories I'm going to tell. One is about glycosylation markers that we've worked out within our own lab. So these are studies within the lab. And then the second one involves much more collaborative work. That's about discovering new kinds of CDGs by whole exome sequencing, which you've heard about already from Sesh Cole, and also the kind of stuff that Eric Green was talking about this morning. So just as a quick reminder, I want to mention again that transferrin as a marker of glycosylation disorders is really very important. You can run this by isoelectric focusing, as many people have done, and I often say you could run this in your garage because it's a very simple assay. But in addition, there are more sophisticated techniques such as electrospray mass spectrometry, which I think is quite a superior technique. And this is a peak, this A peak, is what transferrin looks like when it's normal. You have 2 N-glycan chains that are shown here. And in type 1 CDGs, and you remember Thorsten described that as the early synthesis of precursors and their attachment onto proteins. If you have mutations in those kinds of genes, then you don't have normal glycosylation site occupancy, and that's what you see here, where this peak and this peak arise because there's not enough glycosylation, and each of these sugar chains is missing. Here it's missing 1, and here missing 2 chains. So you'll need that information a little bit later. Okay, so story 1 is we know that transferrin is a good marker for CDG patients, but there's a problem, and that's that there is no equivalent of transferrin for individual cells. So you take cells from patients, transferrin is not found there, and if you put transferrin in, it doesn't show under glycosylation. So that's not very good. Well, and you say, well, do you even need such a thing? And I think the answer is yes. And the reason, again, goes back to the kind of thing that Michael Jackson talked about and that Eric Green referred to, and that's that if you have a marker, it can provide confirmation of putative defects that you may identify through whole exome sequencing or in trying to look for a drug that you can see in high throughput screening. So if you have a marker, then you would be able to use that marker to tell if these things are actually working. So we use 2 approaches to try to identify different markers. And the first approach was one that was worked out by Peng He in the lab, and this is using a quantitative proteomics approach. And so basically what you do is to take some group of cells which are glycosylation competent, and you label them with normal lysine. for about 6 generations. You take another group and you label them with heavy lysine, and this is incorporated into the proteins over several days, and this is in a group of cells called LEC9. They're a glycosylation deficient cell line. So why do you want to do this? Well, the idea might be if you had a problem in glycosylation, that something that's normally present in control is not going to be there because it will be lost, because it won't be stable, because it won't be functional. And you can tell that difference. So what happens is you make these extracts, make membrane protein extracts, solubilize them, do some fairly complicated mass spectrometry, a lot of informatics, and what you look for is the possibility of seeing an underrepresentation of some proteins here in the group that is glycosylation deficient. So this is really important. Well, Ping worked very hard, and he was able to show that the best candidate for this was a molecule called ICAM1. And the idea is, well, if that's really true, let's see if we can really prove that to be the case. So he also looked not only in this artificial cell line, if you will, a cell line that's used most of the time by a lot of people doing biochemical analysis, which was called Chinese hamster ovary cells. But he also then took this to a number of different CDG lines. And he looked at the expression of this protein by immunofluorescence, by FACS analysis, and by Western. And basically it worked. And so what he was able to show is whether you take CDG1A cells, 1B cells, or 1K cells, or even groups of patients that have some unknown defects that we haven't figured out yet. In any of those, you can see that compared to the control, which is done in black here, all of these curves move to the left. And moving to the left means they don't have as much of that material. He then did the same thing by a Western blot, looking at type 1 CDG here compared to a control, and you can see there's an enormous difference, normalized back here to integrin alpha 5 as a control. And the same thing is true if you look at type 1A cells and 1B cells are the unknown. So again, by each of those ways, you can show that I think he's done 25 or 30 different abnormal cell lines, glycosylation deficient ones, that here is a protein that seems to behave in an aberrant way. If that's the case for 1B, we know that 1B patients and cells respond to mannose. And so the prediction is if you put mannose onto those cells, that you'll now begin to recover the expression of that protein. And in fact, when you do it by FACS, here's the analysis with no mannose in the culture medium, and now you go up like this as you increase mannose, and you see that increases by FACS. It also increases in a Western blot as you add more mannose down here. So again, that works. That's a validation of that as a marker. But we also, so that would be a drug. We also want to be able to do this in a way that you can do gene complementation. So we know what the defect is in CDG1A, that's phosphomanamutase. So if we have the gene and then put that back into the cells, the prediction is we'll be able to correct the deficiency in that gene by enzyme activity. We'll also be able to have a correction of the ICAM1 expression. So here, if you compare, is the very low level of enzyme activity in 1A cells, and here's what it should be in control over here to the right. Now, just by putting in the vector itself, you of course don't increase the amount of activity. However, when you put in the vector with PMM2, you get a tremendous increase, and it's even above normal. And when you do that, you can see again by FACS that there is an increase. Here's what it is in 1A, and here it is in the complemented cells. Now you'll notice even in the empty vector, this increases from there to there, and you say, well, what's going on there? Well, it turns out that ICAM1 expression is regulated or can be influenced and increased by viral infection by itself. So that's a control that was really important. So then Pig did the next thing, which was to sort cells that he had corrected into different groups that had a flag tag on them. So we had a flag tag PMM2. So what this allowed us to do is to take those cells that had a little bit of PMM2 activity and flag, that's over here to the left, versus those that had a high amount over here to the right. And what you can see when you do that is that you now get this very nice increase. At low levels, you see very little effect. At high levels, you see almost complete correction. So this was a validation that said you can really use this as a marker. There was another idea too, and that would be that if you could design a green fluorescent protein that had a potential glycosylation site, you might be able to assess whether that protein was glycosylated or not. And so it's fairly common to put glycosylation sites in proteins, but it's not so common in doing this in GFP. And part of the reason is GFP doesn't normally exist in the endoplasmic reticulum where it would be glycosylated. So what Maria Stell-Losfeld did was to make some constructs. And the construct that she made was to put an ER signal to get GFP into the ER and another signal to retain it in the ER. Then she added a glycosylation site. And you say, well, that sounds pretty simple. Well, it wasn't very simple. And the reason is because what you needed was a glycosylation site that when it was occupied, when it had the glycan on it, there was no fluorescence. When the glycan was gone, it glowed normally. So what was happening is it was the presence of the sugar chain itself that was actually destroying the fluorescence. And so what she was able to do with this is to actually show that you can get glow versus no glow depending upon whether that sugar chain is added. Just changing the amino acid at that location doesn't make a difference. So that's very important. So now we have a second marker. And how did that work? Well, it worked beautifully. So rather similarly, what she was able to do is to show that, in fact, as you added tunicamycin, which is an inhibitor of glycosylation, you could go from cells that had no glow over here to ones that had glow over here. And in fact, even if you measure the incorporation of tritiated mannose into glycoproteins, you could show that at very low concentrations, you could get a modest amount of reduction of glycosylation, but there was still significant amounts of glow that occurred. And in fact, if you plot it over here, you can see that even at very, very low levels of tunicamycin, you can get a profound increase in the glow, meaning that the oligosaccharide is not attached to that protein. So she also expressed this, stably expressed it in HeLa cells, and back here are the controls. And now when you add tunicamycin to inhibit N-glycosylation, the glow comes back. Correcting with mannose, the same thing, much as Ping had done. Down here, where you try to add PMM2, which is the known deficiency in CDG1A, you complement that. So now what you would get is the existence of glow to start with, because those cells are glowing because they're glycosylation insufficient. You add the gene to it, and now it destroys that glow, and what you see is the glow coming down here. So it goes from glow to no glow. Well, now we've used those tools to do collaborations with different groups, and one was with Emory Genetics, and this is Liam. And we said, well, what is Liam's defect? And Liam was just published here a week or two ago. And it turns out that he is deficient in one of the oligosaccharyl transferase subunits called DDOST. It's also called WBP1 in yeast, but the oligosaccharide transferase complex. In other words, the complex that actually transfers from the lipid-linked oligosaccharide, puts the glycan on the protein. That's what's deficient. And we got a note from his mom this morning, and she said, well, what is his CDG type? And I said, we'll put it in the talk today. So it's DDOST CDG. So what do we find when we try the ICAM1 marker? Again, it gives us just the thing we would expect. There is a loss of ICAM1 ICAM1 on Western, and also by FACS analysis and by fluorescence. And we also look what can happen when you now put the DDOST gene back in. Here is the level initially with a very low level compared to control, and now we put the gene in and it works. So we have really two independent ways now that we can bring to bear on looking at the gene that's responsible. Now I'm going to go back for another one to the transferrin profile that we talked about before. So you remember that normally what happens is that transferrin gives you a profile like this where two sugar chains are sitting on the protein and it gives you that mass. Now, if you're missing one of the chains, you're going to get this kind of a pattern. That's a CDG type 1. And then we ran into this thing we called a mixed type. And what in the world was that? Well, it had the missing chain like that. That was typical. You know, we had seen that before. But Lord, what is this? And how do you, knowing what you know about biochemistry, cell biology, you're adding a sugar chain in the ER, you're modifying it in the Golgi. Those are two separate ways of going about glycosylation. So how can you find one mutation in what kind of gene that would account for making both an absence of entire sugar chains as well as individual sugars on those sugar chains? We scratched our heads for a long time on that one. That was a really tough one. Clinical presentation. We knew of about 6 different patients, and they had no developmental delay in contrast to many of the CDG patients we've worked with. Many of them had a variable amount of hypoglycemia. There were liver abnormalities and oftentimes a coagulation defect. Another thing that was rather typical is they had a Pierre-Robin sequence or cleft palate. And that was true in about half of the cases that we had run into. And so again, this didn't tell us what the gene could be. Well, turns out that this is a collaboration with Thorsten Marquardt, and that's not Thorsten on the screen. That's Mason. Mason, would you stand up? Would you stand up for us? All right. Thank you, sir. So Mason and his family said, geez, what's going on with me? Well, thanks to what Thorsten had figured out by whole exome sequencing, he identified a patient that had a very similar kind of clinical abnormality together with this abnormal transferrin profile that we called the mixed type. So when we looked at that, we found that in fact, it was a defect in PGM1. That's phosphoglucomutase 1, and that's the enzyme that interconverts glucose 1-phosphate and glucose 6-phosphate. And Thorsten talked about that earlier today. So we found, doing enzyme activity on the patients that we had, as well as doing Western blots, that all of the patients that we had were actually deficient in this enzyme. So there are now, I think, 14 was the latest count for the total number of patients, and this has just come together in a fairly short amount of time. So this is very unusual to, I would say, to discover this many patients suddenly. Once you get a clue, once you see that you've got something in your hands, then now you can begin to look everywhere. And that's what's so important about the collaborations. Because now you can talk to all your buddies, you can talk to all your friends and say, hey, you got any of things that look like this? Yeah, well, we know what to tell you to look for. And that's why this collaboration is so important. You look at PGM1 in terms of ICAM1, again, the same thing, that's working. We noticed that there was a decrease in the sugar nucleotide or nucleotide sugar ratio of UDP galactose or glucose to galactose. What it appeared to be is UDP galactose appeared to be lower. And Mia Ishikawa did some really nice studies on this and showed us that that was the trend. And so what could this be? Well, this is a rather complicated slide, and I won't go into it at the moment, but that's a lot of the biochemistry that goes in to try to figure out where the sugars are going within the cells. But one of the things that we thought is that if the UDP glucose is up and the UDP galactose is reduced, and Mason and all of his friends have a deficiency in adding galactose to these proteins, maybe if you were just able to provide lactose or galactose, that might help. And we thought, well, is there any more biochemistry we can figure out from this? Well, yes, it turns out that glucose 1-phosphate is elevated. So again, this would fit into the scheme that Thorsten described earlier this morning, that would fit in with an elevated UDP glucose, reduced UDP gal. So what could we do about that? Well, we have no idea at this point how an increase in glucose 1-phosphate can cause underglycosylation. In other words, a failure to add whole chains to the protein. We didn't understand that. We still don't. But using Maria Stell's protein, we were able to apply Michael Jackson's technique of high throughput, high content screening. And in the 96 well plates, we had cells sitting in there that would glow. And now these could be photographed, digitized, recorded, and then we could measure the amount of glow in each of the wells. So what Maria Stell did was then to show that you had glow here in GFP, and that fits because it's under glycosylation. And then now if you add galactose, you can see that the glow is reduced. This is just up here on top to count the cell numbers to show that we have an equivalent number of cells in all. And then one thing that Thorsten suggested was to add uridine, because uridine is a precursor to UDP galactose. It comes from UTP plus galactose 1-phosphate, now gives you UDP galactose. And so when we did that and you quantify it, you can see, in fact, that looking across all of this area, and this is just one of the plots, that you can significantly decrease the amount of glow. In other words, you can improve the glycosylation of adding entire chains onto the proteins just simply by giving galactose. So is there anything that we can do with that? Well, Mason, as it turns out, has been on an escalating dose of lactose for the last about 3.5 months, something like that. So his doc has been escalating the dose of lactose being given to him, and that's a prelude to probably moving toward galactose. But the thing I want to point out is here are normal levels of the forms that are -1 chain and -1 sialic acid. These are the baseline levels. And you can see that starting back here when Mason first started taking lactose, that this has been on a steady decrease. Now, we need to go longer. We need to probably take those doses up. But we also need to do this very carefully because this is new territory. We don't know. Could there be side effects? We have to be careful. We do the same thing over here in looking at some of the other peaks that were artificially high in several of the patients, and we can see that they've come down into much closer to a normal range. So that appears to be working, at least on a biochemical level. Now what we have to do is to see over time what Mason's 100-yard dash time is, I guess, huh? So we went to, I'm going to move on to another syndrome that we looked at, this thing called Chyme syndrome. And Chyme syndrome has a set of symptoms that include ocular coloboma, heart defects, ichthyosis, mental retardation, which is called intellectual developmental delay, and epilepsy. So that aggregate of symptoms, was rather unusual, and it was first reported in 1983. In fact, there are only 8 known patients that have been reported on in the world, and we managed to get 6 of those, and this was practically a single-handed study by Bobby Yang in the lab. And so he was able to basically track down this defect based on several aspects, I would say, that many people might have missed. But he looked at the whole exome sequencing, found some extra things in terms of heterozygosity mapping, put it together and figured out that it was a defect, not in a gene that we thought it would be because it looked like our other patients, but it was a defect in a gene called PIG-L. And as of today, Bobby told me he just found yet one more patient today whose DNA had been sent. And Bobby's one of these kind of guys that always goes back and checks, to see what the results are the last 10 minutes, right? So as of, 2 hours ago, I think you sent me a note that the final patient on our list certainly has this. So what is PIG-L? Well, this is involved in the glycophospholipid anchor synthesis, and this is something that many people in glycobiology are very familiar with, but many people outside the field may not appreciate it very much. But there are a large number of steps, and these are anchors that will substitute for a transmembrane segment of a protein. So the transmembrane portion would be pulled off, and at the same time you'd have a transfer of a lipid oligosaccharide that will anchor proteins within the membrane. And so this is one of the steps within that pathway. And there's a number of steps that are now being reported in that pathway. Well, all patients, it turned out, had the same point mutation. It changes a highly conserved amino acid. And the carrier frequency is it's less than 0.1%. And all of the heterozygous patients that have been found have been in Northern Europe. All of the patients where we found deficiencies are of Northern European origin. And they have a variety of different splice mutations and things like this, but this is the one constant mutation in all of them. And we also confirmed that there were 2 GPI-anchored proteins that were decreased in the cells we were able to get from a couple of these patients. So we've got 3 defects that we know about, but we've got some new defects that are on the way. We think we're within striking distance, if you will, of a new OST subunit defect. There's a nucleotide sugar transporter that probably is going to turn up to be one of the next type of CDGs. And what's interesting is we didn't pick that one up by exome sequencing. We used the differences in transferrin to see this. And in fact, for what we know about the genetics, it's very likely that this would have actually been missed by whole exome sequencing. So the combination of biochemistry, that kind of analysis, and whole exome sequencing, genomics, new generation sequencing is going to be essential. What are the rate limiting steps? Well, I think you heard Eric talk about that we're going to need more bioinformatics. And that's going to be really essential for us. We have a number of patients, and I'll show you coming up here, but identifying the candidates from the whole exome sequencing takes a lot of time because the bioinformatics pipeline is complex. It takes a lot of work, and there are a number of people here at our institute and other places who are trying to work on this. And again, I think one of the other things that can slow all of this down is if there is insufficient information sharing among all the research groups. We got to share that kind of information. Once you're pretty certain you've got something, then we have to, everybody, talk to each other. Because look, all of us at one point or another are going to find some new defect. Maybe we find it in one patient. Maybe there are other groups that have more patients. It's in the interest of our employers, the families we work for, to take care of their needs as well as our own. So we have to share as much information as we can. So the current status of our whole exome sequencing project looks about like this. We've got collaborations going at Emory Genetics. We've got our own in-house group that's working on some of this. Eric Eklund, one of the former postdocs in the lab at Lund University, is working on some. And Peter Robinson, who spoke here last year, has several samples. We've just started up a new collaboration with Sessions Cole and Mike Bamshed at University of Washington. And Sesh, of course, is from Washington University. And one thing I want to point out about this is if you go here to the point of Emory Genetics, we have 33 samples that we've analyzed. 4 of those patients have new disorders. Only 7 have known disorders. So 7 out of 33. And the other 22 that we don't know about, we have looked for all the known genes and all the genes that we know are very likely. So about 50 genes, we've eliminated all those. So what that means is in those 22 patients, there are probably lurking how many new disorders? We don't know, but certainly they're not falling into the categories of the ones we already know. There's a lot to do out there, and we hope to keep it up. Where in the world do we do this? Well, if you look across the world, and please excuse the map, and we know Canada is not that small little country up there. We've sort of moved the tectonic plates around a little bit. But you can see that we have a lot of physician collaborators. And when I started off this morning, I said, that's one of the things we want to do. We want to make collaborations among scientists, physicians, and we have to have collaborators, scientific collaborators. And you notice every place there's a green star, that's where we have scientific collaborations going on. We need more over here in Europe. We need more in Australia. We need more around the world, and we want to get those. And the Middle East, I think, is going to be an absolutely great place to get some collaborations, and we really look forward to that. So we got 3 new defects that are in progress. I think a lot of new disorders are going to be found. Biochemical analysis using transferrin pointed to a defect that we would have totally missed by whole exome sequencing. So you got to do that. You got to incorporate that as part of your diligent search for CDGs, because sequencing only provides candidates. You need to have functional and biochemical confirmation, or else you won't get the right answer. So now I want to mention a paper I think that was extremely important that just came out about a month ago. This is out of Christian Kerner's lab in Germany. And basically, he made a mouse using the 2 most common mutations for CDG1A. Now, most of the patients in the world that would be born with these mutations would survive. None of his mice with that mutation survived. And you can see that over here, where he's looking at the offspring, the number of live offspring that have these mutations over here to the extreme right. If you look at 9.5 days post-conception, you have evidence of live embryos, but any time after that, nothing. And there are no offspring that are born. So that is a lethal disorder in mice. So what can you do about it? He was, I hear a suggestion. So he said, well, let's see if we can feed mom mannose. What could possibly happen? Well, how much mannose is a mouse mom going to drink? I mean, do you have to have this like maple syrup, to have it so thick, there's so much mannose in there? No, they gave just a little bit of mannose. And guess what happened? They survived. They survived not only to be born, but then after gestation, those mice could be taken off mannose and they were fine, suggesting that there was perhaps a critical period during embryogenesis where just a little bit more mannose, just a little bit more, was going to be sufficient to make the difference between life and death. We think that that is really a highly significant observation. And it gives credence to the kinds of things that Thorsten has talked about earlier, to all the talk we've had about mannose. There's another side, and we feel it's our obligation to bring this up. And I take a quote from the paper that we really love some of the results. In light of the positive results achieved in this study, initiating a therapeutic mannose regimen to women at risk for CDG1A descendants before conceiving, or in the case of CDG1A confirmed in utero by early prenatal diagnosis, may overcome impairment of clinical developmental steps that influence postnatal development of the disease. We feel that is an overly strong statement, perhaps even dangerous, and Thorsten and I believe that this statement is premature and overly encouraging to parents at risk for having CDG1A children that maybe if you just drank mannose, you wouldn't have to worry. We don't feel that that is the right statement to make at this point. And if anyone thinks about doing something like this, please talk to the physicians who are involved in this. That's very important. Because we know, we've seen, as I mentioned before, blind mice from drinking mannose prenatally. Okay? So this is my list of collaborators. I guess you can even say it's a short list, but as you can see, we're trying to do what we said, which is to collaborate, to reach around the world, to look for different places, and to go out there and find those diseases that we can. And we think there's a lot more to be found, and I think our collaborators will help us find them. We're always looking for new collaborators. I'll stop there.
Have time for one question, perhaps? Go ahead.
So your ICAM1 expression studies in the fibroblasts, I mean, it really looks very clean and can be used for a gain of function assays, right?
We're not doing it yet, but that's one of the ideas, is that you could have a glow come back, yeah.
Oh, it's striking.
Yeah, I mean, it really is pretty amazing, isn't it? I'll tell you, the one downside, and I referred to it, is if you put a virus in there, you know, now it ups the level. And one of the problems is that if you overexpress this, the difference between them goes away. Well, we don't understand it. So it's only, you'll notice with the FACS scans, I mean, they were at the very low end, right? And so the expression is very low, so it's right on the edge, but it works. Yeah.
Very quickly.
Good stuff.
I thought it was intriguing to see hypoglycosylation if you have increased glucose 1-phosphate in your cells. This is a little bit reminiscent of the G6PC3 deficiency, where you have hypoglycosylation of NADPH oxidase genes. I wonder, you said you don't understand yet, but do you have any clue, any hypotheses, any?
Well, I think, yeah. His paper was just fascinating on this. I mean, the bottom line is that when you have glucose 6-phosphate in neutrophils, and you either don't transport it into the ER, or you don't, you don't cleave off the phosphate, so you accumulate glucose 6-phosphate in the cytoplasm. You have the same, I think, kind of phenotype. I don't know about the missing chains. Okay, but that's a really important issue. So I think the thing to remember is now glucose 1-phosphate increase, fructose 1-phosphate increase, galactose 1-phosphate increase, and glucose 6-phosphate increase all give underglycosylation and lack of adding chains. So I think there's a whole segment of sugar phosphate metabolism that is really controlling and signaling that we know nothing about. And your studies and the things we've run into here are the Rosetta Stones that are saying, you know, look in this area because there's something going on. And I think it's got to do with regulating sugar phosphates. And so we're measuring a lot of these in cells by very sensitive enzyme techniques. Yep.
Okay. All right. Thank you, Hat.
All right. Thanks.